Thank you very much, Alejandro. So the discussion is open now. Do we have some questions in the chat? No. So who would like to ask a question? I see a question, raised hand from Bob. Please go ahead.
Thank you very much. Really cool. Thank you very much, Alejandro. Nice work. So yeah, I'm from the biology department, and I heard David Nieuwenhuis also present some of this deep learning approaches, and it seems really valuable. So I was wondering if you use sort of regular genome alignments and look for conserved regions, then would you find similar locations, and would that be much slower, or would it be in another way disadvantages compared to this deep learning approach?
Actually, we went directly into using deep learning because the alignment has the part that there can be disaligned by certain number of letters, the sequences, and this one, it doesn't matter because it works through computer vision. So it only, you input the sequences and it will find a visual pattern and it says, here it is. It doesn't matter in which part. But when we do the explainable AI, when we pass from sequences to the, am I sharing?
No.
Share again the screen. So
the genome alignment in theory also contains the full genome length, right? So it doesn't matter where it also contains the full genome. But I like the.
But in this part, we make our own alignment, but only of the important features. So we take out everything that doesn't matter to actually separate the viruses. And we do this for all of that, which is different from the technique in alignment, because alignment, many times in the beginning, there can be displacements, and some of these features will appear afterwards, even because all of the sequences are aligned when they put it in the repository to start with.
Yeah.
So this is how to compensate all of this. That's why we went to deep learning to apply this instead of using the alignment methods.
Yeah. I still wonder if you find the same, if you would find the same mutation. For example, that one mutation from the Diamond Princess screws, that was kind of interesting because that points to a specific outbreak, like a local outbreak where a lot of patients had the same phenotype, right? Is that correct, my interpretation?
Well, it points to the same location, but not all of the patients had the same phenotype because it was across, it was people from all over the world. So it was like a contained
same disease phenotype, sorry.
Sorry?
I meant the same disease phenotype?
Yeah, from the virus, it was like without, yes, it was the same phenotype of the virus.
So symptomatic, asymptomatic, right?
Yes, it had symptomatic and asymptomatic. So it was like a contained experiment where we could actually separate both very easily, because then you have to move to the global stage, and then we do not, countries, normally they do not measure asymptomatic also.
So was this mutation that you found, was that characteristic of the Diamond Princess cruise patients, symptomatic or asymptomatic, or did it separate those out specifically from that location?
In that location, it's separate. No, okay. No, this mutation in the beginning, we thought it was only connected to the Diamond Princess in the beginning, but that's why we added 2 other mutations to make a decision tree to be able to separate asymptomatic and symptomatic. So the most important one is to check if it was Diamond Princess, and then to apply to all over the world, we defined 2 other mutations in a decision tree to be able to separate between symptomatic and asymptomatic.
Okay, because I'm wondering if you need to maybe use a more phylogeny guided approach, because now every genome sequence that you compare is weighted equally, right, by the machine learning. It is just one observation, one more observation, and if these sequences are actually clustered as an outbreak, then maybe you want to weight the genetic relationship between the viruses into the decision. Anyway.
exactly. No, I understand the question, because in the beginning it was like, okay, we're able to separate the people from the Diamond Princess. Although the people from the Diamond Princess had some properties that were really interesting, because there the death rate was really low, considering that so many cases, because we always hear the 3%, that 3% death rate, although actually the numbers from WHO are 5 to 7, but we always hear the 3%. But this one, it was 1.3, and the people that actually died from the Diamond Princess were people above 80 only, and it was in the beginning. So it has these properties. But to compensate globally, we also find that there were two also important mutations that define the symptomatic and asymptomatic, containing the ones of the Diamond Princess.
Can I
give somebody else a chance to ask a question?
Can I correct you in that? I'm not sure. This is Janneke van der Weijert. I'm an infectious disease epidemiologist. I actually think the death rate on the Diamond Princess was probably normal. The difference with the rest of the world is that they captured all cases because it was a cruise ship, whereas in the rest of the world, a lot of people die in nursing homes without ever being confirmed a case. So the death rate probably is, 1.3%. The 5% is a death rate that they measure in hospitals. So those are very specific cases that tested positive. And even in the Netherlands, at least half of our deaths were never confirmed cases of COVID.
Exactly. So this part of that's why it was necessary to find something else to separate asymptomatic and symptomatic, and not just be able to identify the people from the.
Yeah. And the other thing I should say actually also is that a lot of COVID infected people who don't die are also not identified. So the overall outcome of that process of under detection of cases in general, not just the people who died, but also cases in general, means that the death rates are, really are not reliable.
So, I have a question. So probably the death rate globally is around 1.3%.
Yeah, I think that the death rate in the Diamond Princess is much more accurate than what we're measuring in the rest of the world, because in the Diamond Princess they tested everyone, so they knew for all, they knew their status and they knew whether they survived or not. And that's not true in the rest of the world. In the rest of the world, most cases go undetected, both the ones that survive and also the ones that die. We actually do detect, of the people that die, there is a higher detection rate, of course, because they often end up in hospital where they are tested. But in the community, a lot of people never get tested, even though they had the infection.
I have a follow-up question. Is because there was another cruise, and the other cruise had a different death rate, because there were two cruise ships in Japan. Well, I don't remember if it was in Japan or if it was the same company, but the other cruise has a different death rate. And
okay. Well, they're just samples. They're just samples of the world population. So yeah, were they vastly different though, or were they just slightly different?
No, it was like really 3%. It was closer to 3%, the other cruise. And thinking this part like they are like close experiments, because they are really close, isolated experiments. And they were able to measure everyone, well, almost everyone on board. And they can measure asymptomatics, everything that we need. So why this case of the, how you say, these people of the Diamond Princess and the other one will be different?
Well, I think you're talking about an N of 2, two cruise ships. I don't think you can draw any conclusions based on that. But
I agree.
Based on what we know of the death rates of viral infections, I think the 5% is much too high.
Yeah, the 5% is really high. The one.
Can I ask you about? I thought your presentation was fascinating. Very, very nice work also for us people who are closer to the clinic. Very interesting. I was wondering that one mutation that you found on the cruise, the Diamond Princess cruise ship, that predicted between symptomatic and asymptomatic patients. That's really fascinating. that mutation, in what gene was that? You know, which part of the virus does it code for?
If I do remember correctly, it's in ORF1AB, but then again, ORF1AB is 2/3 of the genome of the virus.
Okay.
So,
I was wondering if it has something to do with receptors, like it attaches better, yes. Or you know, what's the reason why? Yeah.
There's a reason. Actually, it has an implication. Well, this was done by the molecular biologist, and I will just repeat what she said. Okay.
Okay.
It's something that given this mutation actually changes the behavior of the viral infection and actually changes the proliferation rate.
Okay, so it has to do with replication of the virus then.
Yes.
So it's, okay.
Interesting.
That's very interesting. Yeah. Anyway. Yeah.
Okay. Thank you. Do you have further questions, Bas? Because there is someone else raising his hand.
Adrian, I have questions, yes, but let's let Adrian go.
Yeah,
first.
yeah, that's what my point was. So Adrian, please ask your question.
Yeah, thank you. Well, I was patiently waiting, so thank you for giving me the opportunity to ask you a question, Alejandro. It was a nice talk, thank you. Actually, I have two questions. The first one is regarding the way you define the primer. So there are many actually parameters that you have to take into account when you design RT-PCR primer. So GC content, melting temperature. You have to look at the number of GC repeats because otherwise the sequence can make hairpins. And I was wondering, of course, the size of the final product that you're going to amplify, so the size of the amplicon. Did you also encode it in your algorithm, all those parameters, or is it something that you just filtered out after you actually got your hits?
I'm going to share again the screen.
Yeah.
So, yes, this one. So, here. So, to answer your question, so yes, we first find the sequences and we do not encode anything else in the part of deep learning because that part was not possible to encode it in that step. So
that's what I thought I understood indeed. So you were quite lucky.
So, but that's why we kept all of the possible choices that we could use. So instead of just keeping like the one that was put out like the algorithm, we run it several times. So we got 12 possible sequences. Yeah. So from these 12 possible sequences, using the primer 3+, we first check which one could do a primer set, only 2. And then when we did using fast PCR, when we do the simulation of the PCR, the melting and this part that it doesn't repeat in the genome, everything else. But this part that doesn't repeat in the genome from the primer, that part I think that it was encoded somehow because we find specific features that do not repeat in the virus that are the most important. So this part, so it's a little difficult to know. But for everything else, through fast PCR, we were able to design the primer set that actually we tested afterwards. Because if it didn't pass the fast PCR step, it was like, okay, theoretically, it's really nice, but until there.
Yeah, so that's what I thought. And the second question was related indeed to the questions that you had already before. So it seems to me that the difference between asymptomatic or symptomatic, that well, maybe it is hard-coded in the genome sequence, and that's what you were actually showing. But it could well be also, of course, that this is something that is more like a host characteristic, so a human characteristic than a virus characteristic. There are many studies at the moment, many initiatives that are genotyping actually infected COVID patients to try actually to find the genetic variants that could actually discriminate between the populations that are maybe going to have an asymptomatic form of the disease or the ones that are going to have a more aggressive form of the disease. So could you make actually any relationship between maybe the mutations that you found in different virus strains and some human genotype variations?
Actually, that was like that was like my dream data set, to have both the information of the sequence of the virus and also the messenger RNA expression of the patients. But Until now, no one has that. But I can tell you that from messenger RNA experiments between healthy controls and biopsies from COVID-19 patients, we find, for example, the most important gene, expressed gene, to be able to differentiate between healthy and COVID patients was AGTR2, AGTR2, yes, and this one actually encodes in ACE2, which is a receptor that it appears in all of the papers right now that they are the one that trying to block. So people are looking for treatment that modifies this gene AGTR2 to be able to block somehow ACE2, because I do not know this part of biology, but apparently they cannot target directly ACE2, but they can target AGTR2. So it was very interesting for us that we find this meaningful difference through the machine learning. But yes, so it's moving towards that. But right now, I do not have the data. If I had the data, probably, by independent experiments, we are, by independent experiments, we find some correlation. Hopefully, having the whole information, we will be able to find it. Hopefully, I answered.
Thank you.
Okay, thank you. I see another question in the chat by Barbara Vrede. Barbara, could you please clarify your question?
Well, it's been asked a number of times already, but my question was about this feature associated to symptomatic and asymptomatic. And my first instinct, like Adrien also said, is that this is actually whether a patient is symptomatic or asymptomatic is something that is a feature of the patient, not of the virus. But you've clarified already a little bit more on the side of like what is the expectation that the gene in question does, that the mutation in question has an effect on. But I would like to learn a little bit more about how you have sort of ruled out the idea that this could be an accidental correlation, rather than that this is an actual, this is a feature that has an actual effect on the biology of the virus and therefore the phenotype of the patient.
Well, actually, one of the things that we do mention is that this, how can I say it? We are not able to rule it out perfectly. We can only do the statistical tests meaningful to separate, to say, well, we have two different groups. And actually, when we submitted this to the bulletin, they say, please change the title to probable missense mutation has an effect on severity, not just mutation has an effect on severity. They made us add the word probable because we cannot actually be sure until we have the whole data set, both of the patient and the virus, to be able to say what is happening, the interaction between the two of them. The way that we validate in this sense, I do not add the statistical, the rock curve, and all of these things that we do statistically to be able to show, and the MANOVA analysis to show that the two groups are different. So you are right. We need to do this step. This is the next step to say what is happening. All of you are right, because this question has come.
I have a question. So one about this. What do you expect would happen if you took your entire data set of viruses and you just randomly permuted the labels, the symptomatic, asymptomatic? Would you find how often would you find this a mutation this significant in the genome?
So, but when you do this permutation like this is like the p-value.
Yeah.
So we calculate the p-value and all of that was calculated. Actually, we use a multivariate analysis to calculate the permutations, because in this case we were having a vector of features and not just one feature. So this part we know there are two separate groups as far as statistically we know. The only thing that is missing and connecting some questions already is that we need to have information from the patients, because it's the effect of the virus in the patients that matters. It's not the virus itself. So maybe by looking at, that's why right now we're working with messenger RNA expression, but with what we have, not so much information there, to see the expression levels of the genes, because it's easier to understand what is doing the virus there.
But what is the p-value then?
The p-value is always let me see if I do not have it here. Well,
like, is it 0.001 or is it 10 to the minus 52?
It was 10 minus 4, something like this.
Okay.
So 10 minus 4.
Yeah. Corrected for the genome size, because basically every mutation is a potential observation, or not? Or
yes, in multivariate analysis, yes.
Okay. Interesting.
Yeah, because we have a set. This is, well, for the people that are hearing, the p-value is just when you use one feature. MANOVA is when you have a vector of features. So basically, you're saying this vector of features is different from this vector of features when you do the permutation analysis.
Thank you.
Okay. Are there other questions, maybe? Ayub, I see a question. Please go ahead.
Yeah, thank you. Thank you, Alejandro, for your presentation. I'm from Methods and Statistics. I have 2 questions.
Please. I will try to answer them.
Yeah, good. Thanks. So the first one is about the architecture of your neural network. So I was wondering, how did you come up with this architecture? Because you didn't mention that you used a validation set. So my question is that, did you use also a validation set, or you only use tenfold cross-validation? So because if you use the cross-validation, then the test set in the tenfold is used to find the best architecture of the network.
No, in this case, the architecture of the network was something that we defined in the beginning. In a tenfold cross, when I say tenfold cross set validation, was we only run the algorithm 10 times to calculate the accuracy.
Yeah, no, I mean that, so you didn't use any validation set.
No, we do not use any validation set because we have too few examples of one of the classes to do the validation set. And we didn't want to go into making, how you say, artificial information, because we were not so sure about the difference in the beginning. So we defined the system, and when I say tenfold cross validation, the tenfold cross validation, that this only means that we calculate the accuracy as the average of running again the whole training.
Yeah. So then, what would happen if you add another convolutional layer?
We had a very complicated system with 3 convolutional layers and max pooling. That was the first system, but it was too big and the accuracy was too high. So when we were trying to do this explainable AI, we went to as simple as possible. So we do this one. This one only has 1 convolution, max pooling, fully connected, and softmax. Basically, that we could trace back the operations manually. So, yeah. And we got the same results, which were really high. So we say, well, for example, what is the point of adding another convolutional layer? And the idea was here in this one, which is a very simple model because it's only input, convolution, max pooling, which normally they are together, fully connected, softmax, which softmax and fully connected is basically the same. To be able to trace back what the network by itself is giving weight. So to try to do by hand the operations, to go back to the most important features, to try to reconstruct the information in the beginning. You see? So it was try to, even in this point, well, to answer the first question, yes, it is very important to do training, test, and validation. No, sorry, I forgot. No. For this one, we have validation. Yeah. For the convolutional neural network, we have a validation set. Sorry. Yes, we had a validation set.
So the architecture is based on the validation set.
We do not have a validation set for the feature reduction because our feature reduction do not take into account the accuracy to do the feature reduction. So for the convolutional neural networks, we have training, validation, and testing. For the feature reduction, we only have training and testing. Okay?
Okay, thanks. So then I want to ask that we know that deep learning has proven to be good with large data sets. In your case, how can you interpret such a high accuracy? Is it because of your data, or is it because of your method? So I was wondering what happens if you use traditional machine learning methods that you do feature engineering manually.
Well, in this case, we applied traditional methods to do the classification of the different sequences, of the 30,000 sequences, of the vectors of 30,000. We use, well, scikit-learning, we just use classifiers. Those were ones, but also we use, what is the name? The ones that are like, convolutions, but using other
QRNNs?
No. It's not deep learning. It's another technique. Like hard transform, that's wavelets. We use wavelets, and we use the standard from the machine learning, scikit-learning, and we get something like 90%. So they were able to identify. But their point was like, in this case, these sequences are displaced like horizontally. So they are displaced. That's why we thought, why not try something like computer vision or deep learning in general that is able to see something displaced, you see? But I actually think, in comparison, because we saw two papers that use deep learning for viruses, we saw that we think the most important part was this, the coding, how we change the letters into numbers that can be used for the deep learning, because they were using different coding.
Yeah.
Okay. I have a question about the coding, actually.
Yes.
Can I do that, Laurence?
Yes, you can. You have time.
Because the coding, generally for deep learning, DNA sequences are encoded with one hot encoding. Are you aware of that?
Yes.
And so why did you choose for this one?
For this one, is the problem of one hot encoding was that we were not able to trace back the explainable AI to go back, because going forward, we are able to classify going forward using the hot encoding. But going back to do the operations that for us were very important, that we're able to do the primers and the severity, which is the explainable AI, we are not able using the hot encoding. So actually we use this representation that you can see here.
Yeah, sure. But in this one, the G and A, for example, are further apart than the G and the T. Isn't that a little bit artificial?
Yeah, but it works.
It works. Okay. No.
Yeah, so it works. The problem with one hot encoding was that it makes really, really big, and then when you handle the memory and all of that, you are not able to input. For example, if you are going to input for this case, the virus was 30,000 bases. We can input the whole 30,000 bases directly. And if we're going to do that with one hot encoding, it makes it bigger, and we were not able to trace back using explainable AI.
Oh, okay.
Hopefully,
Okay. Thank you. Are there any other questions? We have still some time for questions.
May I ask something, Laurence?
Yes, please. Go ahead.
So, if I understand you correctly, Alejandro, maybe one of the main features of your method that makes it successful is that you enforce the invariance of the sequences being possibly shifted. Let's say if we could do that and then apply wavelets or whatever, then it would work similarly. Is that right?
But I thought that wavelets, because one of the ways that they use to classify viruses, I saw that there were mentioned wavelets. So I asked someone, a friend of mine that works a lot with wavelets, to make the comparison, but they didn't go as far as a convolutional neural network. I thought that wavelet, because wavelet in sense is for this type of things, to find in a signal, to be able to find segments of a signal, which is what we were doing, right? But no, wavelets were more or less the same as the standard from the scikit-learning.
Right. No, I'm just thinking the main thing about convolutional nets is enforcing an invariance, translational invariance in the case of convolutional nets. And it seems to me like that's maybe also tying in with some of the earlier questions. That's maybe the main redeeming feature of why after this convolution you don't need many extra steps. Just dense and move on with life. I think that's an interesting finding. I mean, I wonder how that ties in with what bioinformatics people are doing, for example. It's just a thought. I didn't really have a question. Sorry.
No, but this goes really deep into the theory of deep learning. So it's also like I didn't think about it. So something to think about.
And maybe tying in with your last remark, why wouldn't you be able to trace back sort of heat map type feature explainers for a softmax or one-hot encoded vector? I didn't understand why that wouldn't be possible. I can understand it might be computationally more challenging, but I didn't understand why you wouldn't be able to apply explainable AI to it.
Well, for sure, you can find an algorithm to be able to go back to do the inverse, for sure. But,
right.
But theoretically,
aren't right now.
Yeah,
is that what you're saying?
Exactly.
Okay.
So theoretically, there should be, but practically, for example, this one was once that we do the convolution and we do only the max pooling, we were able to see visually this part, where we were able to see visually some patterns, and we say, well, this is enough, maybe.
Yeah.
And when we extract these features, we get these beautiful patterns that actually we can see the difference between the class, and they say, well, this is enough. We did not go further into what else could we do, and we stop here.
No,
and
sure.
Yeah, but yeah, that's really interesting. So try to do the explainable AI path also in the one-hot encoding, but it's gonna be more difficult because you have to compensate for the one-hot encoding.
Yeah. You'd need to think about how to do that. It's interesting to hear that hasn't been worked out fully yet. I guess there's something still left to do. But very, very impressive work overall. Very, very nice presentation also, Alejandro. Thanks.
Thank you.
Thank you, Daniel, for your question. There's still 2 minutes. So if someone wants to raise a question, please go ahead. Oh, I don't see any hands raised. No questions in the chat. So I guess that we can wrap up then. So thank you very much. Also on behalf of Peter van der Heijden, Alejandro, thank you for your very interesting presentation. And thanks to all participants also for joining this webinar and also for participating in the interesting discussion we had at the end. This video, the video that I made will be placed in this Microsoft Teams channel, but we will also try to place it on our YouTube channel, so it will be available. And Alejandro told me also that he will share his slides as well. So thank you all very much for your presence, and hopefully till the next time. Bye to all.
Thank you, everyone.